now. Okay, so syllabus, here it is, the schedule. Today is the 31st. We'll be finishing up chapter 6 today. I mentioned that several times now. We're a little bit behind in the lecture portion. We're not going to probably get to chapter 12. I'm not worried about it. We still have plenty to talk about and to cover for the test, test 2, which will be during finals week. And by the way, is not cumulative. It is only over the second half of the course. Whoops. Let's see, though. You did have this quiz due, Master's Emissary Primacy of the Right Hemisphere, just now. I did post the Chapter 8 quiz Monday. It is there and ready to go. This afternoon, I will be posting a quiz for the Ancient World chapter of Master and his Emissary. We only have 2 more quizzes for that one left now. You had a quiz due today, and you only have 2 more. And I will mention once again that these last 2 chapters we're going to read, these are where there's a big focus kind of on the speculations of McGilchrist. There's a focus on some of those big picture ideas about how the hemispheric differences influence society and how they've influenced society during the course of their changes over time, their evolution. So starting in the ancient world, which is obviously a long time ago, and then moving to more recent times in the final chapter that we're going to read, Romanticism, the Industrial Revolution, more recent events. There are other chapters in between that we're not going to cover. This kind of gives you like the beginning of what he's talking about, the ancient world, and it kind of jumps to somewhat modern times. There's a couple chapters in between, and then there's 1 or 2 chapters after the Industrial Revolution where it's talking about even more modern stuff. You kind of get the idea from these, and as I mentioned last time, especially if you're doing option 1, it's not a bad idea to kind of browse some of those other chapters to kind of get an idea of what some of those bigger picture speculations are in terms of kind of the other time periods of Western society. It's not required, though it can kind of help you to get a bigger picture understanding of really what's going on and how it's been applied to all of these different points in history, all these different points in time. But once again, I will be posting the Ancient World quiz this afternoon. Any questions or comments about upcoming assignments and things related to the schedule? All right, not seeing too much there. So we'll go to chapter 6 stuff. So there's some hard concepts and experiments in this. So I do want to talk about some of these things in a little bit of detail. And I do want to I think this is where we left off talking about kinesthetic codes. And this is the idea that the dorsal pathway can assist what the ventral pathway usually does. So the ventral pathway, we talked about how that's important for object recognition or object perception, which I have usually decomposed that into kind of like 2 different parts. Object recognition is being able to name something that you're looking at or hearing, but we're talking about the visual system mostly. So object recognition, one part of it is naming something that you're looking at. The other part of it is being able to describe what that thing does. That's kind of the ventral pathway, which relies, ventral pathway, to remind you, also known as the what pathway. It's the one that's the lower pathway, the ones that go from the occipital to the temporal lobe and maybe pushes up a little bit to the frontal lobe. The dorsal stream or the where pathway, that goes from the occipital lobe up to the parietal lobe. That's like the upper or superior pathway. I mentioned that that's also involved with 2 things. One of them is spatial perception. And spatial perception means, more specifically, kind of representing where things are in space so that you can interact with those things. So the spatial aspects of spatial perception, that's the dorsal or where pathway, which makes sense. Spatial perceptions of where are these things so that I can use them, so I can interact with them, so I can touch or grab them. The other thing that the dorsal pathway is thought to do is kind of store your knowledge of how you interact with different types of objects. So I gave the example last time of my magnifying glass, which I ended up putting away. So it's not here anymore, but my magnifying glass is something, the motor knowledge, the behavior that I usually engage in when I see it is when I'll reach for it, and then I'll grab it and I'll put it up to my eye and look through at usually a small object like a coin or something like that. So that's kind of like the motor routine that my brain, that my mind has connected to this specific object. But there's different objects have different type of motor routines. The scissors, which are shown here, kind of the motor routine for them is this motion. And there's another one we talked about last time, I think. Oh yeah, we talked about the combination lock one, which I'll remind you of in the next page. In fact, I'll go to it right now. Yeah, so we talked about this last time. I'll talk about this top one here in a second too. The combination lock person, GS. This is the person that was shown a combination lock. That is one of those things that would be like on the locker, the dial that you turn on a locker so that unlocks it. And he couldn't recognize that when he originally saw it. This GS could not recognize that. Originally, he said it was like the dial for a rotary phone, the old school style of phone from like the 70s and early 80s. But the researcher said, no, that's not right. And then, so he had to keep on trying, keep on thinking that at some point he kind of spontaneously was doing this. This is the motor routine that his mind, even though he was unable to recognize what it was, he was not able to name what the thing does. He incorrectly said it was a rotary dial from a phone. He was not able to say it was a combination lock for a locker. He was not able to name it. He wasn't able to describe what it does in words. So his ventral pathway appeared to be impaired because he wasn't able to recognize the object. However, his motor system still seemed to kind of understand what to do with the thing that he was looking at, because he kind of spontaneously started doing this, which is the motor routine that you would use when you're engaging with the object that he was looking at. So there's this interesting separation of knowledge. One part of your brain is able to kind of describe the object kind of verbally. It's how I usually think about it, the name of the object and what it's used for, often describing that in words. That's the ventral pathway. The what pathway? The where pathway, or the dorsal pathway, that's where there's the knowledge of how your motor system interacts with the thing. And his motor system, his dorsal pathway, his where pathway, was still able to remember how his body interacts with that thing. And he started doing this motion. And then he looked at his own hand. It says use of his own hands to recognize the combination dial lock. And once he saw what his body was doing, then he was able to name the object, combination dial lock. When he was using just visually, he couldn't do it. He was just looking at the thing. But then he got his motor system involved, which still remembered something about what the thing was. And then he's looking at what his motor system was doing. And that eventually provided him with enough knowledge to kind of allow him to retrieve the name and the types of things that object did. He's able to verbalize that information once he got the motor system involved. And so I mentioned last time, this theoretically, what's going on here is that the information in the parietal lobe, which is thought to store, memories of how you interact with objects, that is connected. It is not completely separate. There are connections between the dorsal and ventral pathways. If you are unable to recognize an object with your ventral pathway alone, those motor routines, that information about how you act with an object motorically, that information can be sent down to the ventral or what pathway, and then together they can kind of help you recognize an object. That motor knowledge that you have, the motor system knowledge, can be sent down to the ventral, that is the what pathway, and can help you recognize something. But there's this, we have a question here. The question is a direct message. So you might want to send that to everyone so everyone can see your question. But it says, if he couldn't see the combination lock, then how could he look at his hand and realize what he was doing? That's a decent question. For one thing, I wouldn't say he couldn't see the combination lock. He could see it. His eyes work fine, but he was not able to take that visual information and from what he was seeing of the lock and then connect it to the correct word for that lock. He wasn't able to take that visual information and connect it to a verbal description of what the lock does on its own. But his motor system would see the lock and would still kind of spontaneously become activated. It's like unconscious almost. There's just a part of his body that when it sees the thing, it knows what it's supposed to do. And that engages the motor system. And I don't know exactly how it happens, but it appears to be happening. There's different pathways. One of the pathway, the motor pathway, was successfully activated by the sight of the combination lock, which would cause him to start doing this, and his motor system was evolved. And not only was he looking at his own hand, but he could feel it. You know, when you do this, you can feel what's going on. And this, you've done this before. And if you do this before, it might help retrieve memories of yourself opening up a combination lock when you were, 13 or 14 years old, junior high or high school or whatever. So I wouldn't say that the question, once again, if he couldn't see the combination lock, then how could he look at his hand and realize what it was doing? I will disagree when it says he couldn't see the combination lock. He definitely could see it. He just couldn't recognize and put it into words what he was looking at. Motorically, he still did. His motor system did recognize the lock. It was the verbal system, the ability to describe, that he didn't have until he started doing this, in which case, and looked at it in particular, he could both feeling his own body moving and then looking at his own body moving provided enough information that he was able to recognize the combination lock. So one thing I want to say about this is that this type of kinesthetic code applies to primarily inanimate or non-living things. I think the book tries to make it might make a distinction between animate versus inanimate and living versus non-living. For our purposes, these terms are exactly mean the same thing. Animate is the same as living. Inanimate is the same as non-living. And the idea is that kinesthetic codes, like for the combination lock or for scissors or for my magnifying glass, those exist for non-living things, that is inanimate things. I usually think of them as tools. Those things that we tend to grab in our hands, in particular, which are, like tools are the things we usually grab in our hands, and then we do something with those tools, something somewhat specific. I mean, just another example, like with these types of things, the kinesthetic code would be like opening and closing the paper clip. Those types of tools, you hold them in your hands and you have the motor memory of what that thing, what you do with your body when you're interacting with that thing. And so we have kinesthetic codes for objects, tools, non-living things, inanimate things. And the evidence suggests that we really don't have as strong of kinesthetic codes for living things, animals, people, animate, living things. We really don't seem to have kinesthetic codes for this. And some of the evidence comes from individuals that have agnosia or problems with object recognition. And it turns out that people, when they have object recognition problems, that they're oftentimes likely going to have particular difficulty with animate or living things. Because if it's an inanimate thing, if it's a tool, then you're going to have some type of motor knowledge about that tool. And that motor knowledge can assist you in eventually recognizing what it is you're looking at. But when you're looking at like a human face, or when you're looking at a dog, you don't have, like an animal, you don't have a set of like motor knowledge necessarily. Maybe with the dog, you have the motor knowledge of petting the dog's head or something like that. But in general, the idea is that the motor knowledge for living things, we don't really think of as living things as something that we have like a tool, that we have like a set of motor knowledge that we have connected to that thing. What this results in is that when people have agnosia, usually their impairments are more severe for animate or living things because they can't use kinesthetic codes to help recognize those things. So I'll say it again, agnosia, usually oftentimes the impairments will be worse for animate or living objects because there is no kinesthetic code for most living animate objects. Tools, clearly there's kinesthetic codes for almost every single tool you could possibly imagine. Living things, there's not differentiating between people. There's not really a motor routine that you have associated with one person and a different motor routine that you have associated with another person. There's really no distinguishing motor routine that you have between people like what you have between objects. I think I just saw a question, so let's take a look at this question. Do these codes help work through the tip of the tongue phenomenon? I would assume so. That's a good question. I would assume that that's part of it. Like the tip of the tongue phenomena, which I haven't studied that specifically, but you would conceptualize as far as these ventral dorsal pathway distinction. The tip of the tongue phenomena would appear to be your ventral pathway. Your what pathway is not really helping. It's not working well. You're trying to retrieve the name of an object, but you can't. And it's, it's on the tip of your tongue. It's hard to name. And yes, I would imagine that when you are really struggling to name something, thinking of a tool that you're really struggling to name, then I would imagine if you were looking at your brain, the parietal lobe, which is where the dorsal pathway is probably working really hard to try and send motor knowledge down to your ventral pathway to help you eventually be able to name the thing that you're trying to think of. So yeah, I do think these codes would, I've not read a specific study, so I'm speculating here, but that seems like a very plausible hypothesis that dorsal pathway, kinesthetic codes, motor knowledge, that could help retrieve a word that you're trying to find. When the ventral pathway, the what pathway is not working, it's not helping you retrieve the word, you could use those kinesthetic code, that motor knowledge, presumably to help you. I think that is a very good hypothesis, very plausible hypothesis. And once again, I'll just kind of say one more time, kinesthetic codes, non-living, inanimate things, tools, those have kinesthetic codes. And so even if you have agnosia, usually to some extent, you can use that motor knowledge, kinesthetic codes, to help you recognize the thing. It might be slow, but you can usually get there. With living things, animate things, there's not kinesthetic codes, or at least they're weaker. And so there tends to be more severe impairments for animate living things because we don't have strong motor knowledge associated with those things. And so if you have agnosia, those things tend to be particularly difficult to recognize. Okay, so here is one of the challenging studies for this chapter. This is talked about in the textbook, and I think it's important. And so I actually tracked down the original article, and I I copied and pasted, I cropped out a figure from an article which is talked about in your textbook. They provide a figure in your textbook, but it's different, and I don't think quite as good as this one. There's a lot going on here, and I will break it down. The reason why this figure is good, so this is a Fmri study. And what this Fmri study basically shows is that the dorsal pathway, the parietal lobe in particular, seems to represent, when you're looking out in the world, the dorsal pathway appears to represent actions. That is, actions that you perform on some object, kinesthetic codes, motor routines. The parietal lobe, the dorsal pathway, represents your interaction, or just interactions in general, of a body with an object. The ventral pathway, on the other hand, the what pathway, represents the object itself. And so let's kind of look, we'll break this, I'll talk more about what all this stuff means, but let's just kind of focus in on each of these 4 pictures. These were the stimuli right here, these little pictures, for the 4 conditions of this experiment. And I will mention these graphs right here, these are showing the exact same thing. These graphs right here, they're showing the exact same thing. These graphs here, same thing. These graphs here, same. So it looks like a ton of stuff, but there's actually, and I don't know why they did this. If this were me, if this were my article, I guess it's because they wanted to have time on the x-axis for one of the graphs. But these things are almost completely redundant. So you don't have to, the main ones that I want you to understand are the bar graphs. So these line graphs, they're showing basically the same stuff that the bar graphs are. So I want you to focus in on the bar graphs. And what these yellow bar graphs right here, these are the brain's responses to this stimulus right here, this picture right here. These green ones right here, these are the brain's responses to this stimulus right here. These yellow ones over here, they are the brain's response to this stimulus right here. And these green ones on the very end, these are the brain's responses to this thing right here. And let's go kind of dig in, look into one real quick. So the instructions the participants had in this experiment was to fixate on the center of each picture. So you're showing this picture right here. It's small here, but in real life, presumably it would be like the full size of a computer or television screen. And you are told to look at the center. There's probably a fixation point on the computer screen that we can't see right here. There's probably a fixation point right there. And you're told to look at the center of the screen. And that's important because, and this is one of the things that makes this study challenging for students. I will definitely be asking about this on test 2. And despite the fact that I usually warn students that I'll be asking about this, there's still a little bit higher amount of people missing this question than I like. So you might want to put a star next to this or whatever, because this will definitely be on the test. But when you're looking at this picture right here, an important thing about this experiment, they're focusing on the fixation point. And this experiment relies on kind of this idea that those things in the left visual field tend to go to the right hemisphere. And those things in the right visual field tend to go to the left hemisphere. So right here in the left visual field, this is like a salt shaker or a sugar shaker, you know, something you would have on the table at a restaurant. That's in the left visual field. So presumably this is being processed in the right hemisphere of the brain predominantly. This hand, so the salt shaker, the sugar shaker, this is the object in this experiment. The hand is the thing which is doing the interacting. It is the thing which is performing the action. And so we have kind of those two components. The ventral what pathway component is representing the object itself, the salt shaker. The dorsal pathway is representing the motoric elements of what you're looking at, the kinesthetic aspects of it, the interaction itself, a body that is doing something with the object. The object itself, not to be represented in the ventral pathway, the temporal lobe. The interaction with that object, even if it's not your specific interaction here, you're actually looking at someone interacting, though you could kind of imagine that this is your own hand. The idea is that looking at the hand interacting with an object, that's representing a movement. That representation should be stronger in the dorsal pathway, the aware pathway. And so we want to break down kind of all of these findings because they do basically support this idea. So it's pretty strong evidence that the ventral pathway represents the object. The dorsal pathway represents the motor action or the interaction with that object, the body doing something towards or with that object. Okay, so that's kind of the beginning of it, describing this experiment. Now there's one or two more things I need to say before I start digging into the findings itself. This is another important thing, which I believe, if I remember the way I asked this question last semester, is directly relevant to the question, something you'll definitely need to know to answer the question on the test. This top set right here, these are basically the brain's responses in the dorsal pathway. Now, it doesn't say dorsal pathway. That's why I'm really highlighting this for you. I'm really drawing your attention to it. It says AIPS, which I believe stands for probably anterior intraparietal sulcus. Parietal is kind of the key thing here, though. This basically is brain responses in the parietal lobe. They really focused in on parietal lobe, which is part of the dorsal aware pathway. Down here, the bottom row, this says, big F, little u, big G. This probably stands for fusiform gyrus, which is definitely part of the ventral pathway, the what pathway. So you don't know this, so you might want to write in your notes or, somewhere, commit it to memory. The fusiform gyrus is part of the temporal lobe. Hopefully by now you kind of know the parietal lobe, which is what that P stands for. That's part of the dorsal pathway, the pathway that goes from occipital up to the parietal lobe. That's the dorsal or aware pathway. You'd expect this stuff up here to be strongly activated when you're looking at the thing which is doing the action, the hand. So we're expecting this, which is involved with representing actions. The dorsal pathway should be strongly activated by the hand that's doing the action. This fusiform gyrus, this is in the temporal lobe, which is part of the ventral pathway, the what pathway. These parts of the brain should be strongly activated by the object itself, which is that salt shaker thing. So I think I'll probably go through the data kind of a little, it's a hard experiment, a lot going on. I've already talked about it a lot. I'll probably just go through a little bit now, and then we'll come back to it a little bit, just briefly as a reminder, next class, which I guess would be Monday. So let's just zoom in on one of them right here. So the hand, we'd expect the hand is going primarily to the left hemisphere because it's in the right visual field. And the salt shaker is primarily going to the right hemisphere because it is in the left visual field. So the person, the participant is looking at this image, they're fixating on the center, the hand's going to the left hemisphere mostly, the salt shaker is going to the right hemisphere mostly. And we'll also think about, our hypothesis. So with this hand, what we would expect is that there's going to be a lot of activation in the parietal lobe specifically of the left hemisphere. The hand is performing the action. It's using a motor kinesthetic code. It's doing the grabbing motion, which is a kinesthetic code, a motor routine. It's in the right visual field. So what we would expect is that if we look in the parietal lobe in the left hemisphere for this stimulus right here, we should see a lot of activation in the left parietal lobe compared to the right. We should show, when we're looking at this, we should show more activation in the left parietal lobe compared to the right. And I'll show you that in a second. That's just a hypothesis. The other hypothesis, when we're looking at this object, is in the left visual field. So left visual field projects primarily to the right hemisphere. So what we should see for the object itself, which is that in the ventral pathway of the right hemisphere, there should be a particularly strong amount of activation for this thing, for the salt shaker right here. The ventral pathway of the right hemisphere should show a particularly strong activation to this thing because it is in the left visual field, so it goes to the right hemisphere. It is an object, so it should preferentially activate the ventral pathway. So let's see if the hypotheses that I just mentioned are correct. So we'll start looking up here at the top, and I'll remind you this top part, parietal lobe, part of the dorsal or where pathway. These graphs right here are in the left hemisphere. These graphs are in the right hemisphere. Please note that this picture right here, which is like got the yellow border, it also appears over here, yellow border. This picture right here, which got a green border, it also, the same one appears here, over here. So focusing in on the hand right now, it's in the right visual field. We hypothesized that should mean, so the right visual field means it's going predominantly to the left hemisphere. So in the left hemisphere, there should be a lot of activation in the parietal lobe compared to the parietal lobe of the right hemisphere. That's really important. I haven't written this as a quiz question, but I'm going to verbally give this as a one question quiz question that I want you to do as like an in-class assignment. The question that I'll have for you is to explain why we would expect this hand right here to show more activation in the left parietal lobe than in the right parietal lobe. So that will be a quiz question that I ask you. And I'm going to show you that that is the case, that is what the actual data show. Right here, this is the left hemisphere parietal lobe response right here. This yellow bar right here. It's the response in the left hemisphere parietal lobe to looking at this object. You got to go way over here. This is the right hemisphere, but we're looking at the right parietal lobe's response to this exact same thing. The hand is in the right visual field, which goes primarily to the left hemisphere. And if you look at the height of this bar here, I'll kind of draw a line here. The height of that bar represents the amount of activation in each brain region. This is the left parietal lobe. This is the right parietal lobe when you're looking at this picture, when you're looking at the same picture. And you can see the activation in the left parietal lobe is significantly bigger than it is in the right parietal lobe for the same object. And so once again, the thing that I want you to know, and I'm not going to talk about this study I'll talk about the ventral stuff a little bit for the same picture, and then I'll come back to it next class. Once again, the thing that I'm going to want you to explain is for this yellow picture, the yellow border, the hypothesis was we would expect more activation in the left parietal lobe than the right. Why is that? Why would we expect there to be more activation in the left parietal lobe compared to the right? And I also, one more thing that I'll add to the question is, why would we expect the response to the hand to kind of preferentially activate the parietal lobe, the dorsal pathway at all? What is it about this hand that makes us think that the parietal lobe would be activated by it at all? So I'll remind you of these two. There's basically a two-part question. I'll remind you of those before class is done. Going to the ventral pathway, and then I'll move on from this. I know I've spent a long time on this one, but it is important. It's one of the probably the most difficult, but also very important study in this article or in this chapter. So the same picture now, we're focusing in on the object itself. So the object itself, we would expect that to be represented in the ventral pathway, but because this object is in the left visual field, that means that basically in the right hemisphere, ventral pathway, we should have more activation for that object than in the left hemisphere. Once again, it's an object, so you represent the ventral pathway, it's in the left visual field, so you expect that activation to be particularly strong in the right hemisphere, which turns out to be the case. So once again, here's the temporal lobe or the fusiform gyrus, which is part of the ventral or what pathways down here, this bottom row. This is the brain's response in the left hemisphere, so the left temporal lobe, the left ventral pathway, the left what pathway. This is the brain's response to this thing, to this right here. The ventral pathway in the left hemisphere, it's not strongly activated when you're looking at this picture, which makes sense because the object is in the left visual field, which goes primarily over here to the right hemisphere. So if you look at this bar right here, this is the right hemisphere what pathway's response to this object right here, to this stimulus right here. And if you look, once again, the stimulus is in the left visual field, so it's strongly, and it's an object, so it should be strongly activating the right ventral pathway, which if you look, that seems to be the case. So once again, you're going to be comparing these yellow bar right here to the yellow bar right here. So if you look, the right hemisphere ventral pathway is more strongly activated when the person is looking at this compared to the left hemisphere ventral pathway, which is activated a little bit, but it's significantly less. This bar right here is taller than this bar right here. The reason for that is the object is an object, so it's going to activate the ventral pathway, but the object is in the left visual field. So you would expect that activation in the ventral pathway to be stronger in the right hemisphere, which is exactly what you find. So I will be coming back to this one. This will be a quiz, in-class assignment that I'm going to have you do. I'll come back to it at the end of class. Don't let me forget. Any questions or comments for now?
Could you repeat what you said about the study that's focused on the dorsal pathway, like regarding which hemisphere is more activated and like why?
Well, I'll go a little bit into it. I really want you guys to struggle to kind of try and figure out why. So I'll say a little bit, but I don't want to give it all away because that would I don't want you, when you're coming up with the answer, I want you to have to like struggle a little bit to think about it. I will say a little bit, but I'm not going to say I won't say the why, but I will explain, I will restate the findings. Focusing here, the hand is in the right visual field. And so we expect the activation caused by the hand to be stronger in the left hemisphere, which is the case. This bar right here is in the left, which is the parietal lobe, the dorsal pathway's response to this stimulus, is taller than the right hemisphere activation in the parietal lobe in response to that same exact thing. So when the right hemisphere is looking at this thing, in the parietal lobe, so when the right hemisphere gets information about this hand right here, this hand is going to the left hemisphere predominantly. And we also will say that this hand is primarily going to the dorsal pathway. So I guess that's the part that I'm not going to tell you the why. Why would we expect this hand to go more to the dorsal pathway than to the ventral pathway? What is it about this hand, which is something that would make it so that this hand is we would assume that it's represented by the dorsal pathway compared to the ventral pathway? What is it about the hand that makes it something that the dorsal pathway would represent? That's the thing I'm not really telling you. What I will tell you is that in the left hemisphere, the parietal lobe, the dorsal pathway, represents this hand with more activation than the right hemisphere does. And that's the thing that I will want you to explain why. Another way of saying that is of asking this question would be right here, this bar is taller than this bar. This bar right here in the left hemisphere parietal dorsal pathway is taller than this bar in the right hemisphere dorsal pathway. Another way I could ask the question is, why is that? Explain why this makes sense in terms of what we know about the dorsal and ventral pathway. So that's what I'll say for now. I will repeat the why for all of this stuff next time, but I really want you guys to be forced to kind of think about it and try and figure it out for yourself. Even if you don't get it, I just want an effort. And that's what you'll be rewarded for, just, giving it a solid effort. And then we will talk about it next class. I think we got something else here. Yep, no problem. You're welcome. Okay, moving on. Okay, so this stuff is a tiny bit easier, actually. And especially, I just happen to know the questions that I ask about these concepts are more like definitional on the test. So like the graphs we just show, usually the way I'm planning, at least the way I have in the past done that, is I'll show you the graph, and I basically require you to be able to interpret the graph. So that's kind of like, the graph I just showed you, that's like a kind of like an application, or it's like a critical thinking or like a problem solving type of question. You can't just like memorize things and get the question that I'm going to ask you about that graph. You're going to actually have to have the skills and understand the concepts well enough that you'll be able to interpret the graph when I show it to you on the test. That's kind of like a, as I said, like a critical thinking or problem solving type of problem. Versus, I happen to know that the stuff I tend to ask about object constancy or Q invariance, these terms right here, more definitional in nature. And I usually think the definitional type of problems or questions are easier. They're more straightforward for sure. So I'm not telling you that to suggest that you don't pay attention here, but I'm just saying I'll be going more quickly through this stuff. So object constancy, you can see the definition here. It's the idea that you can recognize that an object is the same, despite the fact that elements of that object change, particularly like the context or the positions in which you see that object, which is kind of similar to this idea of view independence, which I'll show you a study for in a second. I guess I'll go ahead and show you some examples of object constancy. So here is a car or a SUV. It's an SUV or a Jeep. And you can tell it's the same car because even though it's in different positions, orientations. That's the idea of object constancy. It seems really straightforward to me, which is, I guess, one of the reasons I don't feel like it's particularly surprising that our brains can do this. It seems not surprising to me, personally. So object constancy, it's the same thing, even though it's in different positions. Same here with this dog. It's in different contexts. It's in slightly different positions. There, the shadow's covering its behind. There, it's facing the other direction. There's a couch, covering its behind. And you can still tell it's the same dog, even though it's facing different ways. It's in different situations, kind of. You still can tell it's the same dog. And that's the idea of object constancy. Even though you see things and they're in different positions, they're in different like orientations. The people wear different clothes. We're able to tell that there's enough about the object which is the same, despite these changes, that we can still infer that it's the same object. You wear one shirt one day, you wear a different shirt the other day. People are still going to recognize you as the same person because there are some features about you which are invariant that don't change. And presumably object constancy is the ability to focus in on those kind of core properties that don't change, that allow us to kind of correctly recognize an object. And I will mention, when we're talking about this, we're kind of right here, where these kind of are a deviation temporarily from the dorsal ventral pathway thing. I don't necessarily, these are more behavioral things we're talking about now. I don't necessarily right now want you to be trying to spend effort connecting these to the dorsal and ventral concepts. Q invariance is another kind of similar idea. The idea is that you can recognize a given object, even if some of those superficial features of the object are different. And there's some examples right here. The superficial feature, which is weird here, is the color of an elephant. In real life, elephants aren't pink. Nevertheless, you can still tell that it's an elephant quite easily. This is of an apple, apparently, or at least some type of a fruit. And you can tell this even though it's plaid. I think you would call that pattern plaid, and there's no plaid apples to my knowledge in real life. So that would be the feature, a superficial feature in particular, are different here. And then this is the human, even though it's, or it represents a human at least, even though it's, really looks like little pieces of wood or a doll. You can tell that this is representing a human. So you can recognize all of these objects despite the fact that the cues, and a cue is just a synonymous, but it basically means the surface features or the superficial features are different, yet you can still kind of recognize the underlying object. So these terms are kind of similar. Object constancy is more about how like the context changes or the orientation of an object changes, kind of like the position of an object, and you can still recognize the object even if that context or orientation changes. Q invariants are like those surface characteristics, those superficial features that those change, but you can still tell once again it's the same object. So in both cases, it's the idea that you can recognize that an object is the same, even though that certain features or characteristics, contextual elements are different. You can still tell it's the same object, just in different position, or it's the same object with different superficial features. There's more, yeah. There's another example of object constancy. So here I'm showing, this is a study which is talked about in the textbook, I believe. And the main reason that I'm showing you this is because it's a very common paradigm in cognitive neuroscience research. And it also supports some of these ideas about the dorsal and ventral pathway a little bit. But really the thing that I find most important about this experiment that I'm about to talk about is the method that they use, repetition suppression. That's really what I find important. This is a method we haven't talked about yet. It's very common. There's hundreds, if not thousands, of papers in cognitive neuroscience that use this. It's a very common way of studying our brain's representation of different types of things. So the underlying idea of repetition suppression paradigm or repetition suppression experiments is that if you, as implied by the name, repetition suppression, you repeatedly present a stimulus, like an object, to a participant. You're measuring their brain activity. Repetition suppression is the idea that there is a decrease in the brain's response to an object the more it is repeated. The first time you show an object to a person, the brain responds strongly to it, typically. But if I show you that over and over and over again, the brain's response decreases. You can kind of think of it as when the brain is surprised, or when the brain, is trying to learn about something new, it works hard to kind of classify, understand the thing that it's looking at. But after a while, the brain gets bored of the thing. You can think of it that way. And it stops responding as strongly. So that's repetition suppression. The name, I'm kind of always surprised that people miss the question on the test about this, because the name has all of the information really that you need. The idea, though, kind of the punchline of this is, at least in theory, it's thought that the decreased activation might reflect the brain becoming more efficient at representing that thing. The first time it sees something, it's trying to represent it, but it's hard because it's a new thing. But you show it over and over and over again, the idea is that the brain gets more efficient at representing the thing, so it doesn't need to work as hard. There's not as much activation to represent the thing it's looking at. I usually just think of it as the brain gets bored when it looks at the same thing over and over again, and so it becomes less active. But that's not dramatically different than saying it becomes more efficient and therefore doesn't have to work as hard with repeated exposure. So in this study, let's see, do we have pictures of the stimuli? I don't actually have pictures of the stimuli, but they did I do need to look at these graphs though to kind of explain what they did. So once again, in this study, they are looking at the fusiform area. The fusiform areas in the, well, I'm curious to know if you guys remember. You guys remember which of those two pathways is, go ahead and use the chat thing, the chat feature. Which pathway, ventral or dorsal, you have a 50% chance of getting it right. Which pathway, ventral or dorsal, is that fusiform area in? You guys remember? Go ahead and I'll give you a few seconds to do this. Seeing lots of answers. The fusiform area, which I will give a clue now, which will probably give it away. It is, since I see 11 answers. It is in the temporal lobe, the fusiform area. So that should give the answer away. Let's take a look and see how you guys did on this. Ventral, 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 ventral. All I'm seeing so far is ventrals. So looks like you guys are definitely have committed this idea to memory, which is good. The fusiform area, part of the temporal lobe, part of the ventral pathway. Ventral pathway is important with representing the objects themselves. And you can see that in the study, there's the word objects right here. In this study, they're looking at the brain's response to some object, and I actually don't remember what the specific objects were in this study. They're looking at the brain's response to objects depending on whether it's the first time you see the object, so that would be new object. And then you can look at an object when it's new, and then you can see it later in the exact same position. That would be same view. So you're looking at the same object that you saw earlier, but it's in the same position. Or another condition is you can look at the exact same object that you saw earlier, but it's in a different position, a different view. So this kind of maps onto that idea of object constancy. The idea is that the brain, parts of the brain at least, are able to recognize that an object is the same even when it's presented in different views, different perspectives, different orientations or positions. But it actually turns out in this study, there's a part of the brain that recognizes an object as, if you take the same object and you move it, it's in a different view, a different position. There is one of these two brain regions actually processes the object as though it's completely new. It's a completely different thing that it's never seen before. That's kind of the neuroscience punchline. So I'll get to that. I guess I was jumping ahead a little bit with that explanation. So here's the left fusiform area, the left ventral pathway, the left temporal lobe. The brain, you see present an object for the first time, the brain responds a lot. That's not surprising. You present that same thing in the exact same view, that exact same position, the orientation. So I show a pair of sunglasses on trial one like this, and the brain responds a lot. Later on, I present to you again the exact same sunglasses in the exact same position, and your brain responds less. That's repetition suppression right there. Decrease brain response when you're presented with the same thing again. Not surprising. That's just classic repetition suppression right there. An interesting thing though, is what happens when it's in a different view. So let's say here for this different view, I'm showing you the same glasses again, but now I'm showing them to you like this. So a different view, a different orientation, a different position. In this left hemisphere area, the brain clearly represents this as being the same object that it saw earlier, because the brain's response is still small. These brain responses are still small relative to when the object was new. So when I first presented these like this, the brain responded a lot. It's like, wow, I've never seen those before. This is new. This is interesting. But then I show you the same glasses in the exact same position later on. Your brain's like, yeah, I've seen that before. That is not interesting, at least this left hemisphere area. But I then later on will show you the same glasses, but in a different position, a different view. And your brain's like, that's not really that interesting. I mean, it's the same glasses I saw, you just turned them. That's not that interesting. It's still the same old thing. And so I'm not going to respond strongly. That's the left hemisphere area. The right hemisphere temporal lobe has, at least in this study, has a different kind of profile. And you can see it over here, a different pattern. So first, I'll show you in the right hemisphere temporal lobe, the right hemisphere ventral pathway. The first time I show you the glasses, the right hemisphere is going to be excited about it. Lots of activation right there. It's going to respond strongly. Not surprising. Also not surprising that if I later show you those glasses in the exact same view, the brain's response in this area decreases. That is not surprising because that's just repetition suppression, classic repetition suppression. You present the same thing to a person, then the brain's response decreases with more repetitions. However, this bar right here is kind of interesting. So here, I'm presenting the glasses like this now. So this is the same object, but it's in a different view. And if you look at the way the right hemisphere represents that, it represents that as though it's a completely new object. So I want you guys to answer in the text chat area. Why am I saying that the right hemisphere is viewing this as though these are a completely different object that it's never seen before? What is it about the data that I'm showing you, that graph, that's the different view? Why am I saying that in this right hemisphere area, when I show the same object but in a different view, it's to the right hemisphere, it's almost like it's looking at a completely different object that it's never seen before? What is it about the pattern of the bar graph, the data, that indicates that the right fusiform area, the right temporal lobe, the right ventral pathway will view this as though it's a completely new thing that it's never seen before? I'll give you guys a couple seconds there. I see 2 responses. I'd like a couple more than that before I'll give the answer here. Not seeing much, so I'll go ahead and read those. Yes, this one is right. It doesn't look like there is repetition suppression. It says in parentheses, the signal change should have decreased, but it stayed the same. Right. So relative to when the object was new, so the very first time you saw this in the right hemisphere fusiform area, lots of activation. You show the same thing like this, there's definitely repetition suppression. This shows us the right hemisphere is capable, this right fusiform area is capable of repetition suppression when you see the same object in the same view. But if I show you like this, so it's the same object, but in a different view, there's no evidence of repetition suppression anymore. This bar right here, which represents the activation in the right area, right fusiform area, is just as tall as the activation in that brain region when it was looking at a new object. There's no repetition suppression here. Repetition suppression looks like this bar is taller than this bar. But if you look, these bars are not different, not significantly different at least. So there's no evidence of repetition suppression here. And notice how that's different than in the left hemisphere. In the left hemisphere, both of these bars are shorter than the red one. But in the right hemisphere, only this one is. And so this is another example of a hemispheric asymmetry, such that basically what this suggests is, at least according to this study, I haven't seen a lot of other research which is studying the same question. So this is just a single study, so there's a caveat. Basically, this suggests the left hemisphere can represent an object being the exact same object, no matter what the position that object. This is in the left hemisphere, no matter how I turn this or whatever position it is, the left hemisphere appears to have object constancy. It represents this as being the same object, no matter how it's turned. In the right hemisphere, though, the right hemisphere only represents this as being the same object if it's presented in the exact same view every time. You only get repetition suppression in the right hemisphere when it's presented the same way each time. If all of a sudden I change the position like this, in the right hemisphere, it's like, whoa, I've never seen one of these before. And so the left hemisphere seems to have like object constancy. It can recognize an object as being the same object no matter what the view or position is. The right hemisphere, that's not the case. The right hemisphere appears to define an object based on all of the details. This is one object. This is another object to the right hemisphere. At least that's what this study says. It seems amazing to me, honestly. But that's what the data suggests in this study. Looks like we have a question or comment. Let's take a look here. So the question is, so is there no repetition suppression then in the left hemisphere? No, the left hemisphere, there's lots of it. I guess I should define this quantitatively. Once again, repetition suppression is the idea that there is less activation upon repeated presentation of something. And so there's less activation for these blue bars compared to these reds. And see how it says repeated objects? These are the same object being repeated over and over again, either in the same or different view. There's clearly repetition suppression here because the responses of these blue bars are shorter. There's less activation here in the left hemisphere in these conditions than there is in the red. So for both of these conditions, there's repetition suppression. In the right hemisphere, on the other hand, you get repetition suppression for this condition because it's shorter than the red bar. There's less activation here, but you don't get repetition suppression here. So the right hemisphere, we have evidence of repetition suppression here because this is shorter than this, but there is no repetition suppression here because this is the same height as this. So we get lots of repetition and suppression in the left hemisphere. In the right hemisphere, you only get repetition suppression, at least in this study, when the object is presented in the exact same view as it was when it had been seen previously. Looks like we have another comment or question. Yeah, good. No, you're welcome. Okay, let's see how we're doing on time here. Not too much time. Let's see how we're doing. Not too much left to go. No, we still have Agnosia stuff. Yeah, we're not going to get done with all this stuff today. Yeah, so I'll just continue on for a little bit. So I'm not going to say too much more about Q invariance. I just wanted to show you another example. So that previous study was mostly about object constancy. The left hemisphere, once again, the kind of conclusion, it has very good object constancy. The left hemisphere can recognize that an object is the same no matter how it's turned, no matter what the view and position is. The right hemisphere, at least in that previous study I just showed you, appears to not have that good of object constancy. It can only recognize that an object is the same object when it is in the exact same position each time. So that was object constancy. Moving a little bit here to Q invariance. I'm not going to say too much more about this. But this is taken from an article in Science. It's an older one, so it's like almost 30 years ago. But Science is a very good journal, and they were looking at kind of the neural correlates of this. I'm not going to explain the brain findings from this Science article. I just want to show you another example of Q invariance. Remember, Q invariance, you can tell that something is the same object, even if kind of the superficial or the surface features of that thing are different. The object here is a square, and here it kind of has one texture. Here it's got another texture, here it's got another. So their superficial features are different, but in each case, it's a square. In case you're curious, what they were looking for in this study, they were looking for the brain regions whose activation patterns were the same for each of these three different squares. Because in all three of these conditions, it's a square. Some regions of the brain will respond differently to each of these because, you know, some regions of the brain, their job is to process those superficial features, the surface features. Those regions of the brain that process the surface features, they will respond differently to these three things because the surface features of these three things are different. So they wanted to first find out, where are the surface features represented in the brain? You know, the superficial things, that's the brain can represent those. Where does it do it? The other thing they wanted to know is, where are the parts of the brain that can represent an object independently of those superficial features? And the way they do this is they present these things and they look at those parts of the brain that have the same pattern of activation for each of these three different conditions: the luminance condition, kinesthetic condition, and the textural condition. I believe that's what those things mean. There was a region of the brain, I believe it was in the ventral pathway, later ventral in the temporal lobe. There was a region of the brain that responded the same for each of these three conditions. And there are also regions, I believe, there in the occipital lobe that responded differently to these three things. So some parts of the brain represent an object independently of the surface features, but some parts of the brain represent the superficial surface features themselves. And yeah, that's, yeah, it was inferior temporal cortex. So there's the abstract for the study I just read. You can, I'm not going to read it to you. You can take a look at it if you want. I think I got most of this stuff what I just said. Okay, we'll start talking about agnosia memory a little bit here, then I'll call it a day. We'll define these terms, and then next time I need to still remind you of the quiz question for next time, and I'll have to type it out. So I'll need some time. So I'll just talk about agnosia versus memory loss, and then I'll go to the quiz question before I forget. So we'll be talking about agnosia starting now, and then we'll finish it, I guess, next time. Agnosia is basically a problem with object recognition. And it's sensory specific, meaning that each type of you would say like visual agnosia or auditory agnosia. Those are the two main types that have been studied. Agnosia is an inability to recognize an object with one specific sensory modality. So this example right here, this person appears to have visual agnosia because when she is just using her eyes, she cannot recognize this object, apparently. Question mark. She's looking at with just her eyes. She's asked, what is it you're looking at? And in this situation, she is unable to say that the correct answer is, a fork. However, if she's able to use another sensory modality at the same time, so here she's looking at it, but she's also touching it, which I'll remind you would also kind of activate that motor system. In this situation, she is able to say, oh, that's a fork. So this would be an example of visual agnosia because the person is not able to come to the right answer. They're not able to recognize the object with the visual sense, the visual modality by itself. That's kind of the core thing about visual agnosia. You have trouble with the visual system to recognize an object, but you can still use other sensory modalities, in this case, like touch, to help you recognize the object. Comparing that with someone who has general memory loss. That's presumably what this person has. When general memory loss is kind of like the whole concept of fork, or the name of the word fork is like erased from your, the dictionary in your brain. So this person, they obviously still know the word fork. They just can't retrieve that word with their eyes alone. They have to use their motor system and their sense of touch to help them retrieve their knowledge of the word fork, to say the word fork. This individual with memory loss, it's almost as though the memory of fork is just completely erased from this individual's dictionary. When he's looking at the fork with just his eyes, he cannot retrieve the word fork. He's got the question mark there. When this person looks at the fork, touches it and lifts it, he's using at least two senses, sight and touch, still can't retrieve it. So agnosia is sensory specific. You'll have one sensory modality for which you have trouble retrieving object knowledge, but you can still use other sensory modalities to retrieve that knowledge eventually. People with general memory loss, they can't retrieve the knowledge at all, presumably because the knowledge has kind of been erased in a sense. For this person, the knowledge is there. They just can't use their visual system to retrieve it. They can use the sense of touch to retrieve it. People with memory loss, on the other hand, they can't use anything to retrieve it because presumably the knowledge is gone, or the pathways to that knowledge are gone. Are people with memory loss forget the word fork itself? Are they having trouble in referring the word and the object? That's a great question. And I don't actually know the answer. And so I would not, of course, require you to know that distinction on the test. It's hard to really know exactly why. The two main theories would be like, so it's possible that the word fork is still represented somewhere in the brain, but it's possible, you could kind of think of it, it's like that representation has, for whatever reason, been like sealed off. There's no more pathways. Like if the word fork is represented in here, but then it's like closed off, there'll be pathways that like reach a dead end here and they can't find their way inside the part of the brain where fork might reside. So in that way, the theory would be, or the explanation would be, the word fork is still in memory, but none of the pathways that would allow you to retrieve it are active, are working. For whatever reason, those pathways have all died and you can't activate it. That's a possibility. The other possibility is that the word fork has somehow just completely been erased from there is no representation for the word fork anymore. And so you might have, you're welcome if that helps. You might have, retrieval, things are trying to retrieve the word fork from your memory by sending signals, but where the word fork used to be in your brain, it's gone for whatever reason. Either one is equally possible, and I don't know which one. I imagine there's some cases where it's one, where it's a problem with retrieval, and other cases where the representation itself is erased. Good question. I like that one. Okay, so I do want to talk now about this quiz question, which I haven't written. I realized that right before class that I needed to have a quiz question for this. I guess I'll go ahead and write it here in this notes portion, right there, and then I'll re-upload these slides to Blackboard so that the quiz question can be written right here. So I'll just write it. You can leave if you want and then just look at the Blackboard questions. It'll take me about a minute to write this. I'll say it as I'm doing it out loud. In class activity for 3/31/21. Today is 3/31/21. Yes. In class, I talked about how the hand response is stronger in the dorsal, I'll put in parentheses, parietal pathway compared to the ventral, but temporal pathway. I also mentioned that the response to the hand in the dorsal pathway was stronger in the right hemisphere than the left hemisphere. Or I should say stronger in the right, dorsal pathway compared to the left dorsal pathway. Explain why this makes sense in terms of the functions of the dorsal/ventral pathways and the crossing of the fibers for him, and the crossing of stimuli from one visual field to the contralateral hemisphere. That's it. So I'll save this now. Now I will reload those to Blackboard. It is right at time. I think, actually, no, I think I still have 5 minutes, but we'll definitely call it a day there. Anyone have questions about this question that I just put up there?
I have a question. I had written down, I might have just written it down wrong, but I had that you said activation caused by the hand is stronger in the left hemisphere, but is it in the right hemisphere rather than the left?
Damn, you got it. Yeah, that is. I totally got that backwards. It's stronger in the left dorsal pathway compared to the right. Good eye. Hopefully people watch this video to the end. So yeah, that is, yeah, good observation. Yeah. So we're talking about this hand right here. Talk about the hand. Thank you for that. That really saved me. In class, I talked about how the hand, and I'll put in the yellow square. Class talked about how the hand and the yellow square brain response is stronger the dorsal pathway compared to the ventral pathway. That makes sense. I also mentioned that the response to the hand in the dorsal pathway was stronger in the left dorsal pathway compared to the right dorsal pathway. Thank you for that clarification. That's totally right. I don't know what I was thinking. Explain why this makes sense in terms of the functions of the dorsal ventral pathways and the crossing of stimuli from one visual field to the contralateral hemisphere. That is now an actual good question, I think. Any other questions or comments? Thank you, whoever did that. I appreciate that much. In fact, who was that, if you don't mind? Will you just send me a private message if you're still there, whoever did that, because That is definitely a good thing for you to have done. Send me a private message saying that you did that. Otherwise, I am going to, yeah, good. I think
When
I'm good.
will this be due?
It'll be, usually these are due 2 days later. So Friday night. And I'll
Okay.
post that on Blackboard as well. Just take a look on Blackboard. Friday night, 11:59 Friday night.
Thank you.
You're welcome.
Thank you.
You're welcome. See you guys later.
Thank you.